one of the 103. 103 out. Yeah, that guy have a white baseball cap on. Not at the time. But they said they both say mid-20s, kind of light blonde hair, light brown hair. We got him on deck. Looks like
Good morning, good afternoon, good evening. Welcome to our International Plant Breeding Seminar. It is a pleasure to have people from around the globe interested in the topic of plant breeding. Today we have with us special guest, Dr. Peter Cousins from Gallo Wines here in the US in California. Dr. Cousins works at Gallo Wines, specializing in the use of modern biotechnology for the sustainable cultivation of grapevines. Before joining Gallo Wines, he worked at the USDA in Geneva, New York for over 12 years, and he was responsible for releasing 5 grape varieties. Dr. Cousins earned his doctoral degree for the University of California in viticulture, breeding, and genetics, and he has a bachelor degree in biological science from Stanford University. He grew up in a family vegetable farm east of Ceres and studied plant science at Modesto Junior College. So, Peter, it is a pleasure to have you with us. I'm going to stop sharing and give you the floor. There you are.
Okay. Let's see here. Thank you, Carlos. Do you see my slides?
Yes, we do, and it looks good, and you can take it from here.
Looks good? Okay,
Yes.
Fantastic. Thank you. It's such a pleasure to be with everybody around the globe to talk about my favorite subject, which is wine grape breeding and grape genetics, and how we are using grape breeding to improve the quality of the grapes that we grow and the wines that we make here at Gallo. Gallo is a food manufacturing company. We're not a nursery or a biotechnology company. We have our business model by manufacturing beverages, wine and other beverages for people to purchase and consume and enjoy. And we see our piece of the plant material improvement as focusing on the development of new products for the improved efficiency of production and for the improved quality of the products that we're going to sell. So when we are developing a new variety, the intention is that it's going to be cultivated by Gallo for production of its own particular wines and other beverages, not by licensing the plant material for cultivation by growers or by selling the vines through a nursery. So when we're discussing the improvement of the plant material, we think about the way that the vines grow and develop and the yield and efficiency, but we're always focused on the end product, which is improving the quality and the cost effectiveness and efficiency of wine production. And because that's true, we focus on the evaluation of the grapes and the evaluation of the wine in that context. So that's the framing of the business opportunity. Most of the wine that's made in the world is made from a few varieties, and most people probably know all of them. About 8 or 10 varieties are responsible for nearly all of the wine that's made, sold, and consumed in the world. Cabernet Sauvignon, Chardonnay, Syrah, Pinot Noir, Sauvignon Blanc, Riesling, and a few others are responsible for the vast majority. So Gallo is certainly growing those varieties. We're purchasing grapes from those varieties, which are grown by outside growers, and that's true of other wineries also. In this case, in the production of wine, we can blend wine from different varieties to produce one wine. And in the general rule for labeled wine from a particular variety, let's say Chardonnay, is that the wine needs to be at least 75% from that one grape, but it could be as much as 25% from another variety. And because we can blend in that way while maintaining the varietal identity of the wine, we have a lot of opportunity to improve the flavors and aromas through blending and to improve the efficiency of the production of those wines by using our own proprietary varieties. So that's the business context of what we're trying to do. Then we'll talk more about the genetics context here as well. Go to the next slide. Okay. Our basic assumption is that the genetics of the plant material, the variety itself, is a major driver for the fruit composition. We can think of it as simply as if you want to make red wine, it's red in color, then you should start off with black grapes because the pigment in the wine comes from the pigment that's in the grapes. You can make a red wine starting off with white grapes, and it's just more complicated and expensive to do that. There's no particular economic reason that you want to go that direction. Then we have many other chemical compounds which are produced in the grape, not just the pigments, but flavor and aroma compounds, mouthfeel compounds, and of course the sugar, which is converted into alcohol through fermentation by the action of the yeast. We think that if we can understand the genetics piece of the composition of those compounds, then we can drive wine chemistry. So genetics drives grape chemistry drives wine chemistry. Grape chemistry is important in the process of winemaking, and of course, grape chemistry also drives composition or quality in table grapes and raisin grapes and juice grapes and the other categories in which we're in the grape context, we're interested in those genetic attributes. We would like to focus on the compounds which are particularly impactful, to focus on selecting for traits and loci which have a large effect, because that allows us to buffer against the variability that's introduced through the environment and the variability which comes through the management practices. So we know that there are differences from environment to environment, and that the way that we grow the grapes will also influence their final composition, and then that will drive the wine chemistry or the wine composition. We want to focus on those important large loci which have an important major effect on the composition, because those are the ones which are going to give us the quickest, biggest impact in breeding, and which will be buffered against those changes in the environment. In many cases, there have been discoveries in the molecular control, the genes which are responsible. In some cases, we even know the specific sequences which are responsible for conditioning quality and yield traits in wine grapes. But there's an enormous amount of discovery to do. We'll talk today about how we're using molecular markers in breeding, and we're in the process of discovering or evaluating our own germplasm for traits which we think will be particularly useful in our new varieties as we move forward. The grape genome is not large, and most of grape varieties are functionally diploid, but we still lack a lot of understanding about how particular sequences relate to the differences which we observe in the grapes themselves in the vine, and also in the final wine chemistry. The output of our breeding program is new varieties, and we now have 3 of our own proprietary varieties which are in commercial production. You can read about them at the U.S. Patent and Trademark Office website, and I'll just briefly describe the attributes of them. We call the varieties 1, 2, and 3, not because we're going to use those names on a label for a wine, but because we expect that these varieties will chiefly be used as differentiating tools for blending to add quality and provide a special benefit to the consumers who are enjoying a Gallo wine in which this grape is one of the ingredients, but may not be the basis for that entire wine. EJG1 is a dark black grape. It has about 3 times the color of Cabernet Sauvignon. Positive aromas include violets and roses. EJG2 is an aromatic variety, but it doesn't have the aroma of Muscat or of Sauvignon Blanc or of Niagara. And instead, it has a distinct sweet herbs aroma. Frequently, people who are drinking the wine say that it reminds them of mint, and there are other sweet herbs which it evokes in consumption. And EJG3 is a lighter black variety, not quite as dark as Cabernet Sauvignon, and it is differentiated by very positive rose aromas. Rose petals, rose blossoms, rose petal candy, rose water are frequent descriptors for this variety. So we're very excited to see these new varieties, which have come from our own breeding program and launched at Gallo Winery. We have additional selections, of course, throughout the full stages of the evaluation and breeding process. Will you know as a consumer that these varieties were used in blending? It probably won't be marked on the label. Instead, when you drink the wine, you'll say, this is really a wonderful wine. I love the rose petal aroma, or I love the minty aroma. And I don't know how they do it, but this is really a great value, and I will buy this wine again. And in that way, we're using our own special ingredients to make wines for consumers to enjoy. Grapevines are a vegetatively propagated crop, and in the course of breeding, we are creating new combinations which will then be propagated vegetatively. So there was at some point in the distant past an original Cabernet Sauvignon seedling. We know by DNA testing what the parent varieties are, but nobody precisely knows when the first Cabernet Sauvignon vine was planted from the seed. And then all of the Cabernet Sauvignon vines in the world today descend from that original Cabernet Sauvignon vine vegetatively, either from cuttings or from grafting. And in our breeding project, we are creating new combinations by hybridization and by crossing among varieties and among our own selections. We work with a commercial nursery to cultivate our seedlings. We don't have our own greenhouse because we want to focus on the characterization of the grapes and the wines and not focus on trying to be a nursery. We make crosses every year and plant the seeds in January. It takes them about 7 to 10 days to germinate, and the seedlings that you see in that picture are about 3.5 weeks old. In the spring of the same year in which they're planted, about 14 to 16 weeks after the seeds are planted, we're ready to transplant the seedlings into the vineyard. And we plant them into the vineyard about 1 meter apart, and then they're trained in a unilateral cordon. They all point one direction, and that allows us to very easily understand which of the shoots in this densely planted vineyard are the ones which belong to which seedling and the corresponding fruit sample. Fruit evaluation is the biggest focus of our work, and we evaluate the seedlings in populations based on their fresh fruit flavor, because there are compounds that we can taste, which we may not be able to identify analytically, but we can taste them. And then we also conduct chemical analysis of the fruit, looking for important impact compounds, and we'll talk more about that in a moment. We plant the seedlings in 1 year, and they are in fruit 2 years later. So the seedlings which are in fruit for the first time, they're in full crop for us in 2025. Were planted into the vineyard in 2023. They grew in 23. They grew in 24. Most of them tried to flower in 24. Well, we want to have a full crop, so we remove the flowers in 24, and then they'll be in full crop in 25. We evaluate in 25. We evaluate in 26. By the end of 2026, we have identified the selections which are the most promising, and we will propagate using cuttings from that original seedling vine. We make 10 vines from cuttings. 10 vines for us are enough to make a small batch of vinification, which is about 30 to 40 liters. Ordinarily, we have plenty of grapes, more than enough for that 30 to 40 liter small batch of vinification. But in the event that we wanted to make 2 small batches, we can do that. We can get a yield estimate from a 10 vine set, which does not provide a perfect prediction of what the final vine yield is going to be, but it's much better than what we can see from that individual seedling. These are some of the attributes that we use for evaluating those individual seedling vines. These are real examples, and what we have are characterization, which happens in the vineyard, and there's a picture of the fruit from the individual seedling. We have some comments which were made by the people who were evaluating the fruit in the vineyard as they tasted it. The first person who does the evaluation of our seedlings each year is an intern. Usually a person who's finished their bachelor's degree and works for us in a 6-month paid internship. And they're the first ones tasting the fruit. So every week during the harvest season, I'm going through the notes of the intern to see what they found, which is exciting. If it looks good in the vineyard and it tastes good in the vineyard, then we can decide to harvest it for chemical analysis in Gallo's own grape analysis laboratory. Here are 2 of the examples of observations we make. We have an anthocyanin measurement, and we have a soluble solids measurement. And you can see that those numbers aren't the same from year to year, but they're in the same general territory. So 1.2, 1.4 milligrams per gram. It's about 20 to 30% more color than Cabernet Sauvignon at this particular location. We characterize the cluster of the plant by taking pictures and measurements. Those take place in the laboratory. There's a fairly large cluster and large berried selection. Cabernet Sauvignon would have a berry weight more like in the range of 1 to 1.2 grams, maybe a little bit bigger. This cluster is quite large. Cabernet Sauvignon would be more like a 200-gram cluster, and Chardonnay more like a 250-gram cluster. So very promising for the potential of yield. We use DNA markers to characterize this particular individual. We know that it is self-fertile because we've made an observation of the flower type. Grapevines have three basic flower types. The one that we want is self-fertile. There are also pistillate and staminate. So this one is self-fertile. It's carrying pistillate, and the fruit color is black skin, red flesh, and it's carrying white. And we can diagnose those attributes using the molecular markers. With all of these many observations that we've made in the vineyard and the laboratory, then we can decide to propagate this vine. We did decide to propagate it and then planted it out into the vineyard at the 10 vine scale. And here is an example of the vine, a picture taken at the time of harvest last year. So is it precisely the same as that individual seedling? No, but just as we saw that it had fairly large clusters of fairly large berries and pretty good color for our location, although we can get a lot darker than this. This is pretty good. And then we can taste the fruit, and I agree that it still tastes yummy and delicious. And this is one of our elite selections that we like as a candidate for red table wine production for Central California. We're thinking about this for the production of really dry red table wines, wines which are not sweet. So that's our basic process for the selection and the promotion of those wines to the level of being able to evaluate them at the research wine scale. So from 30 to 40 liters, we can conduct the analysis of the wines themselves. So first we conducted the grape analysis. That helps us to predict the wine composition. Now we've made the small batches of research wine. We can evaluate the wine by tasting it, by smelling it. Gallo has its own sensory research panel so that we can compare the new varieties against commercial kinds. And we can also use them in blending to see how a new selection may improve the composition of a commercial product. You can make wine in a very small volume. You can make wine in a bottle cap. But the challenge with very small batches of wine is that it's difficult to extrapolate the results and to translate them to the commercial scale. In our research winery, the control over the conditions and the experience of the people conducting the research winemaking is very high. And even though we're making a fairly small batch, 30 to 40 liters, we can use the results and the practices of the winemaking at that scale to inform the commercial winemaking. So once a selection is commercially planted and it's being grown on a large area, then and being commercially harvested, then the question is, well, which yeast should we use, or what fermentation conditions should we use? And those will already have been worked out at the research wine scale. We can simply explain to the commercial winemakers what practices they should use to make it. Of course, they may decide to vary conditions in order to produce a different style. And that's very appropriate since at the small scale, we don't have an unlimited quantity of grapes for making different fermentation conditions, harvest decisions, et cetera. So what about other attributes? We think about disease resistance in a way which complements the wine quality and grape yield and grapevine yield and efficiency. We don't think that consumers are particularly interested in the disease resistance of a variety. They're mostly interested in the quality of the wine and the value, the quality compared to the price. And so we would like to incorporate disease resistance and disease tolerance when it also complements our goal of producing wines with excellent value for our consumers. So in California, the disease for which we spray vineyards chiefly is powdery mildew. And you can see in this picture, difference in disease response based on the REN1 resistance allele. We don't have a no spray vineyard So we are using molecular markers to help us predict this trait. And we focus on the REN1 resistance allele because the marker is pretty good and it comes to us from a Vitis vinifera background, which already has fairly high fruit quality and good vine yield and adaptation to our conditions, in contrast to some of the unusual wild species, which although they do have strong disease resistance, they're not well integrated into elite backgrounds for our conditions. So we work with the REN1 resistance allele, and we have some other powdery mildew resistance types that we're working with. Our biggest focus in the plant disease area is for sour rot. You can see the berries in this cluster, which are discolored, so they're pinkish to brown, and they're starting to collapse. And that is caused by a combination of yeast and bacteria. It's called sour rot because the bacteria are acetic acid producers, and so the aroma is of vinegar. In our conditions, sour rot does not require precipitation or rain on the fruit during the growing season. And in most parts of California, there's very little rain on the fruit during the main part of the season. So for us, that would be from May to October, and we have very little risk of other bunch diseases. But sour rot doesn't require those to become activated, and instead, we see sour rot mostly in plant material which has very tight, compact clusters. We can breed against tight clusters by having clusters which have smaller berries, the clusters which have elongated rachises or more rachis internodes, and they're not as compact. So we don't see the berries being damaged by cracking as the berries are growing in size. If we do get the cracking, then the yeast and the bacteria which are responsible for sour rot are ubiquitous in vineyards. There is no chemical control that we can use to manage those yeast or bacteria, and it's not practical, and there's nothing labeled. And so we can, however, choose plant material which has loose clusters, and that's very effective. So I have to say that coming from mostly work in rootstocks before I started at Gallo with the Grape Breeding Project, I was surprised how quickly we were able to select for very low sour rot incidence, even from varieties which have measurable sour rot. You will almost always find seedlings that have a looser cluster and which are, for our purposes, sour rot free. So that's a big deal because sour rot is really harms wine quality, and there's very little that we can do about it from a plant disease management perspective. So the plant material is a big guide for us. We are not truly selecting material that's resistant. Instead, we're selecting material which avoids the infection of sour rot. We've talked about DNA marker applications, and it's an important part of our breeding project. This is a picture showing our sampling in the greenhouse. We plant the seedlings, and about 3.5 weeks later, then we collect the tissue samples from those individual seedlings. Ordinarily, we're collecting from the tip of the cotyledon. We send our samples out to a commercial laboratory which provides the DNA extraction from the leaf sample, as well as running cast markers for us. And we have validation for cast markers for important traits like fruit color and aroma, such as monoterpene content, and flower type. So we're very interested in cell fertility. We like the platform that we have, and we like working with a commercial partner so that we can focus on the parts that we do well, growing seedlings, making crosses, characterizing the fruit composition and the fruit quality with our own internal chemistry lab. And then we can work with a partner that has very excellent high throughput DNA extraction and also running those cast markers developed by that company, but then validated in our own populations. We use molecular markers for other attributes like looking at parents from germplasm, helping us to design those crosses. And then also, if we have new or unusual plant material, we're trying to understand how it relates to other varieties. So again, that case, it would be microsatellite markers. For fingerprinting, we're working with a different partner, but always working with a technology partner to get to the science question by working with experts. One of the traits which is very important to us in wine quality is the color, and the color of the wine is chiefly driven by the color of the grapes. Most of the black grapes, which are used for making red wine, like Cabernet Sauvignon or Pinot Noir, have the anthocyanin pigment produced exclusively in the skin of the berry. However, there are a few varieties, chiefly from one lineage, which produce anthocyanin pigment in the pulp of the berry also. This is not the leakage of anthocyanin from the skin. Instead, the anthocyanin is produced in the pulp of the berry, the grape, the individual fruit of the grapevine, and we call it a berry. And you can see in this picture an example of black skin red flesh grape. There are many varieties which have been developed for black skin red flesh, and they come in 3 different lineages. We know that the general genetic cause is a tandem repeat of a 408 base pair region in the promoter of the MIB transcription factor, which drives UFGT in the anthocyanin biosynthesis pathway. So Franco Ruckel at JKI in Germany published on this work, and we find that his discoveries and description of this trait is a very, very good description of what's happening. We know that from his work, there are these 3 lineages, and they differ in the number of 408 base pair tandem repeats. It could be 2, 3, or 5. So we had the question of, if you have these different lineages, is there a difference in the average color content of the seedlings from a particular lineage? So Ruby Red and Da Capo and Pallas are varieties which do differ in the number of tandem repeats, but they also have differences in their other breeding. So Ruby Red is an interspecific hybrid. It's about 7/8 Vitis vinifera and 1/8 Vitis rupestris, developed at UC Davis as a port wine variety, and now chiefly used for producing food coloring for foods and beverages. Da Capo is a fine wine variety developed in Germany, 100% vinifera, has a different lineage and breeding because of the different applications, developed at a different location, a different climate, etc. Ruby Red and Da Capo and Pallas at our location all produce around 5 times the color of Cabernet Sauvignon in the fruit, even though they have different numbers of this tandem repeat. But our question is not, are there differences from variety to variety, but rather, is there a difference from the different lineages if there's something on average is different? So that's one of the questions that we had. And we also, of course, want to know what's the average value of this black skin red flesh trait. We can breed for it. We have a molecular marker which predicts the trait, and the molecular marker predicts all of the alleles with the same accuracy. So we're very pleased with that marker, but the question is it valuable for us? Here's an example looking at some populations and we have chosen these individual plants based on their fruit color, and we've sorted them using the molecular marker. So they're all black fruited. Some of them have red flesh and some of them have white flesh. You can see the range in color and you can see the mean. We're very pleased to see that the black skin red flesh trait really, in this case, gives us a doubling of the anthocyanin content of the fruit. there's a wide range. So there's an overlap of the black skin white flesh ones with the black skin red flesh. It's not that every black skin red flesh plant has more color than all black skin white flesh seedlings. But in this particular sample of hundreds of seedlings, which are candidates for selection, we can see that just by choosing the black skin red flesh marker, we would expect to see an important enrichment in the overall color of those seedling selections which we're making. So the cluster weight is not materially impacted by this trait, and neither is the berry weight, although those are also important to us. It does not have an important effect on the total of the color. So we're interested in the color, and that's what we can see, is that we see an important enrichment in that direction. Here you can see some seedlings in populations which were developed to look at the copy number specifically. So if we don't have access to the original clone, which has a 3 copy, but we created 2 copy population and a 5 copy population, and the plants from the 5 copy population have significantly more color on average. So we looked at the plants from these populations 2 years in a row and evaluated their color in each year. Some seedlings from individual populations have more color than a different seedling from another population. We expect that. But we want to know what the average value is. If we're creating breeding populations, which are the ones which are the most likely to have the attributes that we want? And in the case of color enrichment, 5 copy color is going to be the one which is the most likely to give us the highest color overall. The alleles which cause black skin red flesh have pleiotropic effects on the plant itself, and you can see in this picture taken at the end of the growing season. So for us, this would be October or November at our location, and the individual seedlings which will have black skin red flesh will also have red leaves. This is the end of the first growing season, so no plants have fruit at this stage. They're planted in the spring. This is their first fall, and you can see that we haven't used molecular markers to select them, because if we do use molecular markers, you'd expect to see full red color throughout the population, showing that we had effectively enriched for the trait of interest. The 5 copy allele also has a pleiotropic effect, which causes the production of anthocyanin inside the wood. I don't have an application for red leaves in the fall, and I don't have an application for anthocyanin inside the wood, but it is interesting to see the plant biology of what this trait is doing. So we have lots of questions about the biological control, the biochemistry of anthocyanin in the plant, because it's so important to us from a quality perspective. This picture shows Erin Logan, who was our intern a few years ago, formerly an undergraduate student at University of Southern Mississippi, and she did a lot of the characterization of plants in a segregating population designed to look at the inheritance of color coming from a wild grape called Vitis labrusca, selection Alba. We work with Erin in the vineyard and with Margaret Worthington and her team at the University of Arkansas to characterize this trait. Ordinarily, we think of the presence of anthocyanin in the fruit, black fruit, as being a dominant trait. In this case, we've crossed 2 grapes which have low or no anthocyanin, Muscat of Alexandria on the right and Alba labrusca on the left. We could discuss about what color we think the Alba grapes are. Certainly they're not black. We could say that they're bronze, or that they're red, or that they're mud or pink. We're not really totally sure what the color is, but not black. We crossed those together, and all of the seedlings are black fruited, just like the 16-128 or 52 selection that you see in the middle. We attribute this to a complementation effect, and that is that the Alba and Muscat of Alexandria do not have their low color or no color due to mutations in the same gene. Instead, mutations are in different genes. And when we cross them together, we are complementing those deficiencies in the anthocyanin pathway, and we recover black in 100% of the seedlings. Thanks to Margaret and her team, we know that the Alba labrusca has a mutation in the GST4 anthocyanin, part of the anthocyanin transporters, which is similar to bronze 2 mutation in maize. This is the same mutation that's known from muscadine, and even though the Alba doesn't have a relationship to the muscadine grape, it has a different bunch grape, Uvitis 38 chromosome background, but it's a mutation in the same gene. So here are some examples of grapes that are not black and would be appropriate for the production of white wine. We're interested in understanding the relationship of color with flavor and aroma profiles because we are currently locked into a relationship in which red wines made from black grapes remind people of black or red fruits, blackberries, boysenberries, black cherries, for example, and white wines made from white grapes remind people of yellow or white fruits, peaches, nectarines, apricots, pears. Those aren't just associations which are made by looking at the wine when you're drinking it. In fact, if you analyze the wine composition, red wines have different aroma profiles compared to white wines. Why is that? We are not tasting or smelling the pigments. Anthocyanin at the level that's present in wine does not have a distinctive aroma or flavor. So why is there a relationship of red wines and white wines with particular flavor and aroma profiles? And another way to ask that question is, where's the white wine that tastes like black cherries? And we don't know the answer to that, if that such a thing is possible. By understanding the anthocyanin pathways, the genetics of that, and the different germplasm, which is responsible for conditioning those traits, then we may have a better understanding of how to make new differentiated kinds of wine. So in this case, we can see that we have the typical Vitis vinifera. This is one of our white fruited selections, and it is missing the functional B. So B is conventionally that transcription factor, the MIB. In the case of the muscadine and the labrusca, which have a similar fruit color, we could call it bronze. They have a mutation in what we call the A gene, that's the GST4. And then we have a different accession. This is a Vitis mustangensis, which was collected in Texas. There are other white-fruited Vitis mustangensis known, but we don't know whether or not it has a mutation in the A gene or in the B gene or in something else. And we'll determine that by making crosses and testing complementation, just in the same way that we did with the labrusca cross. So, of course, we're interested in pigments, but we're interested in other attributes as well. Here's a population. We see 2 individuals, full siblings, that are segregating for a wax attribute on the fruit. You can see the shiny one and the wild type one. This is best characterized by Justin Lashbrook and his team at Stellenbosch, and it has something to do with the triterpenoid pathway. In this particular population, we're also looking at the relationship of zygosity of the DXS1 locus with regard to fruit composition. So we can use our marker that predicts what's happening at the DXS1 locus, part of the monoterpene biosynthesis pathway. We can think of that as the major locus for the muscat character. Muscat is a lineage. Muscat grapes are high in monoterpenes like linalool and geraniol, and Moscato is a wine style, not a varietal per se, but a wine style that's made from these highly aromatic kinds. And in this case, we have a population that's that segregates for homozygous muscats, those which have two copies of the functional muscat allele, and those which are heterozygous. So we're expecting that all of these, if you made wine from them, or if you fed them to grape breeders, they would tell you that these are muscat kinds. We wanted to know what the impact of the zygosity of that trait is. So should we be selecting for homozygous muscats or heterozygous muscats? The DNA marker allows us to unambiguously identify which ones are which, and we can. And so in this case, you can see that there is a strong difference, a statistically important difference, and a fold level difference in the average performance based on parts per billion of these important monoterpene analytes, geraniol, linalool, nerol, and alpha-terpineol, which is expected. These all come from the monoterpene pathway. They relate to one another. Beta-zamacinone is a norisoprenoid. Even though many muscat varieties are high in beta-zamacinone compared to non-muscat varieties, we don't see any predictive value of these markers for beta-zamacinone. And that is also expected. So it's a different chemical pathway for the production of, again, important volatile chemicals, but it's not intrinsic to the muscats, and it's not a part of the monoterpene pathway, and it's not significantly different. Also, even though we know in other populations that black muscats and white muscats in the same population differ in their average color, there's no apparent influence of the monoterpene pathway itself on the production of color or polymeric tannins. And that's also important, because if we thought that we were going to drive up the level of monoterpenes in black grapes, we'd like to know if we're taking a hit for anthocyanin or polymeric tannins conditioning mouthfeel. We're not, and at least in this population, and that's important to us from the decision-making process about what to choose, what to keep, what to promote in breeding. This is an example of the segregation of linalool across a population. Our first cut is to ask which ones of these plants carry the muscat allele. And you can see that there's a very large range of the non-muscats, which don't have the allele, compared to the ones which do. We know that the muscats have more, but there's still a large range in each category. We're able to use an additional marker, which is the Gewürztraminer marker. That's a different allele at the DXS1 locus. We have a marker that helps predict that trait also. And you can see that although there are just a few, that those help to account for at least part of the range that we see for the non-muscats. So Gewürztraminer are aromatic, but they're not muscats. And although we don't have a DNA marker that we currently use, we do know from parentage analysis that several of the plants in the population or in this collection of seedlings are aromatic muscae seedlings, and that they have an important increase in the average content as well. So we can use these attributes to look for the seedlings that we like, so the ones which are the highest in linalool or other monoterpene compounds. And we can also use this to look at the collection of seedlings and ask, are there individuals which have unusual high positive levels of linalool and other monoterpenes, but which aren't muscat, which are not Gewürztraminer, and which are not aromatic muscae? Are there other mutations in the pathway? So we know from other plant systems that there are major switches which drive up the production of monoterpenes, a couple of genes at the base of the monoterpene pathway, which particular positive alleles tend to increase the total content. And so we're interested in understanding which plant material is responsible for that. I will just describe in closing some ongoing work which presents a bit of a mystery to us as we move forward in grape breeding. That is the aspect of the genetic control of soluble solids. For the most part, as we look at populations, and here's an example of wild type population, we see normal distribution for soluble solid content across the seasons. So it starts off low and then it gets higher. And at any given particular point, some seedlings have more soluble solid content and other ones have less. But they go up across the season. We have segregating in our populations a slow ripening trait in which the soluble solids content is not normally distributed. It is evocative of bimodal distribution, and this trait is important to us as a call out because the attribute for wine is that we're using the sugar for making alcohol, and wine is an alcoholic beverage. So we would like to know the genetic control of sugar, and we know that certain varieties are better or worse at producing sugar for their particular conditions. And here's a case of something which is a large effect locus, as yet unidentified, which really delays our ripening. I'm going to stop there. I can gladly talk about grape breeding and genetics for hours and hours, but I know that we have a particular time here, so I'll hand it back to you, Carlos, for the questions, and thank you so much.
No, thank you, Peter. It's been an amazing presentation. I'm gonna tap into some of the questions that we have in the chat. If you want to stop sharing so we can see you in the full screen. Okay.
Yes, there you go. Perfect.
Is there much variation in the flowering time of those cultivars and breeding populations? That's a question from Lucia Vignali.
If we look across a germplasm collection, like in our location, which we have our research vineyard near Madera, near Fresno in Central California, I was in the vineyard yesterday, and we had vines which were in flower. And I don't know that I caught them on the first day, but this would be typical that we would get the very, very earliest flowering in the first week of April. And we will have other varieties which are at the same location, which are not beginning to flower until the second week of June. So we can have a very extended period for flowering. Within the population, we should say that the rate of flowering, the rate from first flower to last flower on an individual seedling varies according to the location. So upstate New York has a much faster flowering from first flower to last flower, either on a cluster or on the entire vine compared to California. But when we're speaking about California, we would expect to see 7 to 14 days difference in a segregating population from first flowering to last flowering.
Okay. Now, question here is whether in your first generations you already graft the seedlings, or is it on the second testing that you do? When do you introduce the rootstock?
Yeah, great question. We do not routinely graft any of our material in the research program. It's only grafted when it goes into commercial production. So why is that? We find that grafting is complicated, and that it is harder to find somebody who is interested in making lots of small batches of grafted vines than it is to find someone who's interested in making lots of small batches of rooted cuttings. We don't do our own propagation, and we don't have our own nursery, and so we work with a commercial nursery partner. And you could find somebody who would be interested in grafting for you to make 100 small batches of small grafted vines. So we usually propagate 80 to 100 selections per year. So you could find someone to do that, but a large commercial grape nursery puts their small orders at the back of the season. And the effect is that if we were to graft our selections, we would have a 1-year delay in the time that it takes us to get the selections back to fruiting. So does the rootstock have an influence on wine composition? Not very much, maybe a little, but not nearly as important as the scion. So we accept that there will be differences in the vine when they're grafted. Mostly they are improved, they're bigger vines, and of course the rootstock provides protection against the pests and diseases which live in the soil. So at our site, we use a systemic insecticide for managing the phylloxera and the nematodes of the site so we don't graft them in the research setting.
Good. Question from Jennifer here in Florida is great work, but are there similar efforts put into breeding unique strains of yeast for the fermentation process?
Gallo is evaluating yeast. There's a lot of yeast work that happens in companies. There are many, many different strains of yeast which are available commercially. And so Gallo wants to know, if we buy yeast from one company and we buy yeast from a different company, which one of those is going to do the best job, for example, of fermenting to dryness. So completing all of the sugar fermentation. There is yeast breeding and genetics work happening in universities, like at the University of Bordeaux and other institutes. We know that there are differences in their efficiency, in their tolerance of alcohol, their tolerance of different fermentation conditions.
Very, very interesting. Peter, I have a question on my own. When you have these denominations, okay? So what you're developing is a better Cabernet Sauvignon, for example, or Cabernet Sauvignon is one genotype and it cannot deviate from that.
Right. So Cabernet Sauvignon is one genotype. So if we think about Cabernet Sauvignon as a variety, that means the plants must have been grown by cuttings or by grafting from the original plant or from descendants of the original plant. So there was one original Cabernet Sauvignon vine that came from a seed. The same thing is true of Chardonnay. The same thing is true of Sauvignon Blanc. When we have a new kind, a new variety at Gallo, it can't be Cabernet Sauvignon, but we could blend our new kind into Cabernet Sauvignon wine. And the general rule is 75% Cabernet Sauvignon in the bottle. We can still call it Cabernet Sauvignon wine for marketing purposes. That's the general rule. And there are a few cases where it has to be 85%. There are a few cases when you can be as low as 50%, but the general rule is 75%. So we could add in, for example, if we're making Cabernet Sauvignon wine, we could blend in 20% Pinot Noir, and the consumer would only see Cabernet Sauvignon on the label.
So that's very interesting, because I mean these are all genotypes that have been developed a long time ago, and supposedly they may be vulnerable to diseases or current or future. So if you're forced to use 70% to call it Cabernet Sauvignon, how about having something that is 70% of the genetics of Cabernet Sauvignon? Can you call it Cabernet?
Interesting idea, but that's not the way that the regulators have accepted the naming. Yeah.
Okay. Do you see changes in berry characteristics after grafting into different rootstocks? There's a question from Rodolfo.
Not very much. The rootstocks mostly influence the size of the plant. So if there's something which influences the size of the plant, it could be the rootstock itself. You could also have a similar effect with water. So just by putting more water on the vines or more fertilizer. The rootstocks don't have a strong driver on fruit composition itself, right? So they don't have a strong influence on berry size. There might be a little bit. They don't have a strong influence on color, maybe a little bit. So most of the ways we think about it is the rootstocks really are controlling root behavior. Certainly, they influence the uptake of minerals from the soil, which makes sense because that's where the vine is getting the mineral nutrients. Obviously, water and so what would influence the anthocyanin content? Let's say the pigment content of the fruit. Well, the pigment isn't made in the roots. The pigment is made right in the fruit, right? So it makes sense that the scion variety, the fruiting variety, is really the major driver for fruit composition.
Okay. All right. Have you done any work on low chilling requirements?
Again, great question. And in our vineyard, we do not have low chill conditions. So even though it's pretty mild where we are in the San Joaquin Valley, we do still get a winter. Grapes don't have a very, very deep requirement for chilling compared to some other plants, but they ordinarily grow better when they have a winter chill. I would be surprised to find out that anybody in California is using hydrogen cyanamide or Dormex for wine grapes. Certainly it's a part of the table grape production practices in California and other parts of the world. Because we have such good cultural practices for low chill, there hasn't been a lot of work on low chill in grape, although we do know that there are differences in the eco-dormancy, maybe not so much in the endodormancy. I would refer people to Jason Londo at Cornell University for that excellent work in that area.
Well, Peter, thank you so much for an excellent presentation. Very, very interesting. Appreciate your sharing all that information. Thank you, everybody, for attending Peter's seminar. We have a seminar coming next week by Dr. Dilip Panti, Breeding Tomatoes for Foliar Disease Resistance, and same time on Thursday. So thank you so much to everyone attending again, and you guys have a great weekend, okay? Thank you, Peter. Bye-bye.
Bye-bye.
Bye.